This is Andrew from FX Medicine. We thank you so much for your support over the last 2 years. We'd really love to remain clinically relevant to your practice. So if you know of an expert in some area, please let us know. You can contact us on fxmedicine.com.au, Facebook or Twitter. This is FX Medicine. I'm Andrew Whitfield-Cook. And we're at the 2016 BioCeuticals Research Symposium. Right now I'm talking with Dr. Andrew Heyman. And Andrew's got a rather remarkable and varied career. And I'll ask you, Andrew, can you explain a little bit about your career, because it's quite diverse?
It is. I'm an integrative physician by training. That means that I went through conventional medical school, and I'm a family physician. So that part is standard. But what's, I think, different about how I entered this world is when I entered it. Oftentimes, practitioners, physicians, let me be biased in that sense, they typically go through their training, and at some point in their clinical career, they realize there are limits to what they can do in terms of just standard drugs and surgery. And there's sort of this notion of, how can I learn more? How can I better help my patients? And they might sort of stumble into these older therapies or integrative therapies in a sense. That's pretty standard. My background is a little different than that. I entered the integrative door sort of proper when I was 17 years old. And so I was introduced to traditional Chinese medicine, and I was a first-year student in university at the time. And I found a training program just near my school, and I immersed myself in Japanese shiatsu and traditional Chinese medicine. So most of my instructors were from Japan and China, and I was 17 years old at the time. I wasn't sure what I was going to do with all of it, but I knew that I loved it, and I knew that it was important for me personally. It probably would be important for me professionally, but there wasn't necessarily a really clear direction, but I knew it was important. And most of my instructors didn't speak much English. Right. I certainly didn't speak any Japanese or Mandarin, but I had a very authentic Eastern-style education. And the kind of core training that has stood me in such good stead, I think, for the rest of my professional life was because of the focus on being present, being aware, and valuing the simple pillars of health: movement, breath, touch, food, sleep. All of these ideas that form the foundation of the oldest healing traditions in the world, and yet we sort of skip over them as physicians. We don't really have good training in those areas, and that's where I was grounded. So I think for me, it's very different with respect to how I approach patients, how I think about them. And so my challenge is in some ways being an interpreter, translating the languages of both sides of that discussion and debate about how do we approach health and healing in humans. And there are different ways in that regard. So I have the great and deep benefit then of being fully immersed in traditional healing methods for years before I ever became a physician. And then since then have worked at a very sort of high academic level, thinking like a scientist and a researcher. So in that sense, I've had to bridge the gap of what we know empirically and what we can measure. And that's a fine line to walk.
You say a couple of interesting things that raises some ideas in me. And one of the things that I heard you speak about in your plenary lecture was that you learnt about patterns in Oriental medicine, that they focus on patterns.
Yep.
Modern medicine focuses on clusters of symptoms, but are we looking at patterns, or do you see a correlation or contrast? Where do you see modern medicine either being similar to Oriental medicine or totally disparate?
I would say in its sort of classic difference, they're totally separate in the way that they look at the patient and assign meaning to information. traditional healing systems, by definition, are pattern recognition based. And in conventional medicine, if you want to call it Western medicine, we start from the supposition that our power comes from our ability to peer ever more deeply into the uniqueness of a body system or process. So the cardiologist gets the heart, and the neurologist gets the nervous system, and the immunologist gets the immune system, and so on and so forth. And that's a model, it's a concept in a sense. And it allows us to move ever more deeply into understanding sort of a biological reality of a disease process. But the great intellectual challenge is, how do we put those pieces back together? How do you get a cardiologist to talk to a psychiatrist, to talk to a gastroenterologist, and so on and so forth? That's enormously complicated. And to bring a biological reality to that also demands a kind of biostatistical and mathematical modeling. So there's this tension now in medicine between the older way of kind of what I would call silo-based medicine, everybody's in a silo, and then this newer kind of systems biology approach, which is a whole different way of thinking, but interestingly better maps and reflects onto a pattern recognition based approach that looks and feels a lot like the traditional healing methods. And maybe what systems biology will eventually be able to do is to measure and codify these kind of patterns that humans express and bring meaning to what's been observed for thousands of years. That there's a healing going on in medicine in a sense.
You wear many hats, and one of those is the Director of Integrative Medicine at the George Washington State University. But you also are in practice.
Yes.
So I've got to ask you, how do your peers look at what you do? And indeed, what exactly do you do?
It's a good question. I think it depends on which peers you ask. You know, what I've seen is that the field has changed dramatically since I entered. In a formal sense, when in the mid-90s, after having done shiatsu and sort of these traditional healing methods for a while, I was invited to run a big research center in alternative therapies for heart disease at a major university, University of Michigan, which is considered, you know, a premier research center. And so I immediately went into this mode of, you know, how do we better evaluate these therapies? And there are all sorts of research challenges to just asking that question. At that time, most of my peers were shocked and surprised and even potentially disappointed that such an institution would even allow this kind of research to happen. And now, fast forward, and I, you know, direct this program at the George Washington University, and we have a sort of fully formed and growing and even relatively mature platform now for integrative medicine. We confer higher degrees, a master's and PhD. Our graduates now qualify to sit for this new board of integrative medicine. It's in the United States considered its own specialty now. Now that's only a year old, but that's new, right? But it's amazing that that's occurred. And so it means then that you can have kind of a peer-to-peer discussion on a level that we couldn't before. So there's a greater acceptance of the power and value of an integrative approach to care that's happened as a result of, you know, public interest, but also a cultural shift in medicine. It's changed a lot, even in the last several years.
And so into your clinic life, what do you specialize in?
So
do you specialize?
You know,
because that was a pretty broad talk where you encompass a lot.
I have to say, what you find as a practitioner, if you're doing these sorts of therapies, there are lots of doorways that you can walk through to start learning how to apply these approaches. It could be herbs, it could be acupuncture, it could be hormones, it could be detox, it could be lifestyle with nutrition and exercise or mind-body therapies. Most people pick and choose, you know, which door they want to move through.
Patients don't tend to choose.
No, they don't. They're optimizers, right? They're very utilitarian. But a practitioner has to choose. And usually what happens is when you first start, you have a narrow skill set and you attract a relatively narrow patient population, usually relatively well. And they have some basic health challenges that haven't fully resolved through a conventional standard approach. And everybody gets excited because we can see those patients improving. Patients are delighted they found a practitioner that's thinking outside the box. The practitioner's delighted because they see their patients improving in ways that they didn't through the standard approach. But the problem occurs when the word spreads. Because the person that you just helped with some stress and weight loss and nutrition brings in their sister with rheumatoid arthritis and depression and, cancer. And all of a sudden, your patient population starts to shift and become more and more complicated. And you wake up one day and you have a practice like mine, where there's no filter at the door. There's no sort of description of what we take and what we don't take, because we sort of accept all patients at all states of health and illness. And I joke that my patient population is like the island of misfit toys. So it's all the throwaways. Yeah. They've already been to the major universities, they've been to the local specialist, they've tried even various natural therapies, and they're looking for an even deeper approach to try and unwind the depth of their illness pattern. And it's amazing when you get it right. So it's hard for me today to say, what do I do? I would say first and foremost, we have a long discovery process to try and really identify what's wrong with this patient, what's the cause of their problem. And then the sensibility component is we would prefer to use more natural therapies and lifestyle interventions and self-management. But I certainly go through a lot of prescription pads throughout the year. But diagnostically, as we categorize patients, I don't necessarily focus more so on one or the other, although I could say that I may have a predominance of very complicated patients due to Lyme and other biotoxic exposures. That forms the core of the research that I do, and we use it as a model of how do people who contract those chronic infections become so severely ill. So that would be one area I would say that sort of sets our clinic apart.
Something you said just before about uncovering the layers of the onion, if you like, of chronic disease, and you've just spoken on the HB axis and the adrenals, quote unquote.
Yeah.
And the important message that I got out of that is something that I've known for ages but twigged just now, and that is my love for American ginseng. And I've always said, it's a mind ginseng rather than the other ginseng. They're all working on the mind.
That's right.
So when you're talking about therapy and we're talking about stress, how do you, let's say the word ratify, how do you bring it all together when you're talking about, do I look at psychological support, talking, counselling, doing tests with regards to depression or anxiety scales, versus physical type of interventions, whether I give them cortisol, whether you give them cortisol as a slight buffer, whether you are very cautious about using that because they're as an infectious agent that's causing this sort of symptom picture. How do you ratify that all together or choose?
I think you have to start from the perspective of, do I really understand what I'm looking at? You can start from, are their cortisol levels low or high? But that doesn't really tell you ultimately what the cause is. So you have to go to the next level. We do that because we offer special structural MRIs of the brain, so we can very precisely measure key centers such as the hippocampus, the prefrontal cortex, the caudate, the putamen. We want to know, is it a brain that's been under assault? And what's the pattern that we identify? We then also go to the immune system. We look at both the innate immune response, we look at the adaptive immune response, we look at vectors and other immune threats, because that's such a key part of the discovery process. And once we get through all of that information, we can say, this looks like stress that sort of damaged your response, or nope, this is an immune threat, and we're just looking at a reflection of that in the cortisol pattern. But we really want to feel confident that we know what we're dealing with first. And that's where the therapies really shine in that regard.
Are we at the point yet where we can measure the trends, if you like, or indeed the activity of the immune system? We can measure neutrophils, we can measure eosinophils, but do we track them? We tend to have a snapshot, certainly in orthodox medicine, How do you differentially look at that to say, hey, listen, there's a real problem with a chronic infection going on here? What do you do?
We use a set of very specialized immune markers on both the innate and the adaptive side. So I look at the alternate complement pathway, C3A, C4A. We also look at transcription factors such as TGF-beta1, which drives Th17. We look at neuropeptides such as vasoactive intestinal peptide or brain-derived hormones such as melanocyte-stimulating hormone or antidiuretic hormone.
Yep.
We want to know the full complement of the impact of an immune system that's dysregulated. And we track those markers every several months to ensure resolution.
Right.
We now are going to the next level and running genomic studies and pairing that with the patient's proteome, if you want to call it that,
right.
To look at where are their predispositions based on their immune patterns. And so this is the next level of complexity because we're seeing things and the way the immune system is behaving and the damage that it's occurring that we don't understand yet. And I'll give you an example. We know that Lyme as an entity will trigger an immune response. And in the genetically susceptible, that immune response will persist even if they clear the infection. One of the consequences of that chronic inflammation, and we just published a study on this, is that it damages the brain in very specific ways. That these are not just immune markers that reflect an immune system that's not returning to baseline, but that in fact, they are the same markers that are injuring key centers in the brain. What's interesting is that when you take a patient who has the same symptoms as Lyme and the same immune response,
Yep.
for example, a mold patient, they look identical in their proteomic and immune pattern, but the injury to the brain is different. We don't know why they're different. There are other immune patterns that are occurring. There's likely a genomic contribution as well
Right.
that's leading to the uniqueness of the injury. So when we do our special MRI scans, we can tell when a patient has had Lyme, when they've had mold, when they've had a traumatic brain injury, when they've had stress, when they've had MS.
And you can differentiate.
And we can
between these.
differentiate that based on the MRI. The core of all of those processes is neuroinflammation,
Yep.
right? So it's a brain on fire. But why are the injury patterns so unique to the exposure when the symptoms and the general immune response is the same? There are pieces to the immune system that we're not capturing, and there's clearly a genetic contribution that we need to better understand.
What would you say to practitioners then who see this inflamed brain and might be enthused by some of the work of David Perlmutter, Alessio Fasano, and say, okay, get them on a gluten-free diet? Doesn't necessarily look at immune issues.
Right.
So what caveats would you tell them to say, hey, listen, it's great, but don't forget the...
Yeah.
other
I would say
messages?
in the way that we approach those issues, certainly from a Dr. Fasano or Dr. Perlmutter, they focus a lot on food and the way that food might trigger the immune system. What we've found is that especially in the 20 or 30% of patients that have a genetic predisposition to this kind of chronic inflammation, you really need to more fully identify all of the immune triggers first and deal with them before you even get into food. That in fact, that's a third or fourth or fifth step. That you have to begin to modulate the immune system and decrease the microbial load or make sure the patient is getting out of the moldy environment or decreasing their stress, whatever is primarily inflaming the brain. And then once we have that under better control, we'll go to food. But we see food as actually a secondary or tertiary exposure in our patterning. Now, that doesn't mean that they don't have digestive issues and problems. They will and they do.
Sure.
But we don't see that as a primary problem in our patient population.
But they're sick.
They're absolutely sick,
by
and
the
we
time
still
they
need to
come
get the
you.
gluten out of the diet, and we still, we need to do all of that. But we don't lead with that. And we don't lead with it because what we have found clinically is patients don't feel much better when they clean up the diet.
Right. Because
We
they've
have
got
to
so
do-
much other
they
stuff
have
going
so
on.
much going on.
You need a rescue first.
That's right.
Right. Now, you have particular interest in mold issues, chronic mold exposure, which is named CIRS.
That's correct.
So can you explain that? How does it differentiate from the inflammatory response from other causes, if
Sure.
you like. Yeah.
Yeah, what it really gets to is the basics of the immune system. There are really two arms to it. There's what's called the innate side, which is the primary early response. It's the thugs of the immune system.
Yeah.
It's macrophages and neutrophils that rush to the scene of the crime, and they start producing inflammatory proteins that not only create inflammation itself, but also call in the rest of the healing response. Mm-hmm. What's supposed to happen, though, is a handoff from this early innate side to the other part of the immune system that's called the adaptive or acquired. This is where the memory cells start to play an important role, what are called T cells and B cells that produce antibodies and allow the immune system to upregulate more quickly the next time there's an immune threat. What happens, though, in a mold patient is that for genetic reasons, they are stuck in the innate side of the immune response, the unthinking side. They become very, very inflamed, but there's no handoff, and that's for genetic reasons. So they don't produce antibodies. They don't have the regulatory component of that other more sophisticated side of the immune system, so they remain chronically inflamed, but it's within the innate response. That's very different and unique than other states of inflammation. So for example, the kind of inflammation we measure for cardiovascular patients, IL-6, TNF-alpha, PI1, fibrinogen, uric acid, these standard markers, they are not reflected within the innate immune response. And also the diseases on the adaptive side. Yeah. So when we lose immune tolerance, when the body loses its ability to determine self from not self, and we see the development of autoimmune diseases as a result, that's problems in the adaptive side, problems in the smarter side, because that side has lost the ability to determine what's me and what's not me.
Yeah.
So that's where most of medicine focuses, right? That's where rheumatology focuses. That's where problems related to cancer occur, where the body has lost its ability to surveil and repair. Mold and Lyme and other biotoxic threats are uniquely situated within the innate side of the immune system. Very different. And we know in particular that innate immune activation leads to injury to the nervous system, that's the part of the body that takes the biggest hit, and for us in particular, the brain. So even when you have a patient where you've cleared the Lyme, you've removed the mold, they can not only remain inflamed, but they continue to damage their brain. And you have to fix that central nervous system piece as part of a whole therapeutic process to get them better.
I could ask you 5 questions in one go here, but let's talk about therapy. So you need to fix the brain.
Yes.
How do you go about that?
So we know 2 things have to happen first for that to occur. The first is you got to remove the threat or exposure. Turn off the stress, clean up the mold, kill the bugs, whatever you need to do, and clean up the gut, fix the diet, modulate hormones. There's a whole series of steps that we have to go through to make sure that we're turning off the immune system. So the first is deal with the exposure, and the second is return the immune system back to baseline. And we do that through special lipid therapy. We use phospholipids and butyrate. We use specialized forms of fish oil and fish oil extracts. We certainly modulate the immune system through plant sterol extracts and other natural products. But once you get through all of that, you're still left with a brain that's damaged.
Right.
And we have a few tricks that we use in that regard that we've shown in pre and post studies that repair the brain.
Neuroplasticity?
Neuroplasticity.
Hey.
So there you go. So one of them is an extract of ginseng. It's a Chinese ginseng.
Right.
And it turns out that there are very biologically active components of ginseng called ginsenosides. And there's the RB line, RB1, RB2, RB3, and then there's the RG line. And RG3 in particular has been shown to be the most neuroprotective. It decreases inflammation in the brain and it induces repair. So we use that as a nasal spray. We also use nicotinamide riboside, which is a B vitamin derivative that's also been shown to be protective for the brain. And we finally use something called vasoactive intestinal peptide. This is a peptide that's been known about for years, but much more recently it's been identified as maybe the most potent vasoactive regulator. And what it does is it improves blood flow to the brain and it allows adult progenitor cells to do their job more effectively. So we see repair. That's a key feature for us. So we use the RG3, we use the nicotinamide, we use VIP spray, and there are several things as well, other things as well.
What about you mentioned that you said Chinese ginseng. What species is that?
So that would be just a Panax ginseng.
A Panax ginseng, right. So when you're looking at, say, the use of Panax quinquefolium, which has not reversed, but a changed profile of the RG to RBs, do you find
not as effective
use of that? No.
Yeah.
So it tends not to be used for the immune system, or tends not to be as useful for the
brain.
mind.
It's better for.
Are we talking structure here?
Yeah,
or?
We're talking structure.
And fats. You're talking phosphatidylserine. Choline you use.
Yeah, in particular.
Yeah.
So that's the main structural component for the myelin, which is, the coating to nerves, as well as every cell membrane in the body. And we know that when you have innate immune activation, not only does it injure sort of the superstructures of the brain, but it injures every cell membrane in the body. They become sort of shaggy and worn and not efficient at drawing in nutrients or excreting waste. So we use things like butyrate to clear out very long-chain fats or renegade fats, and then we add phosphatidylcholine.
Butyrate, particular bitter taste.
Yeah, it's not fun. No one likes butyrate.
Except in butter.
Except in butter. That's correct. So ghee and other things.
Can I ask you a question about the toxic molds?
Sure.
So one of the common ones that's mentioned in the US literature is Stachybotritis chartarum, which doesn't appear in the Australian literature. Not that there's much. It appears, though, that we have different species causing this toxic building mold issue.
Yep.
What can you tell us about the toxic building mold? Why is it such an issue? Why aren't the building practices that good? What can we do about that? What do patients have to do about it? Is it down to moving?
That is, I think, the core question for people who have mold sensitivity. And let me be clear, in conventional medicine, we are typically taught that mold exposure can lead to an allergic type response. It can also lead to a much more serious pulmonary infection.
That's right.
But only recently in the literature has it been recognized that it can also be a trigger to the innate immune system.
Right.
That idea has not quite caught on yet.
Yeah.
And it turns out about 20% of the population has a genetic predisposition to this sort of mold sensitivity. We know in the United States, up to 50% of US buildings have toxic mold. And the problem is, if you're genetically predisposed, wherever you go, you can get sick.
You can carry it, yeah.
So even though we can get you out of that environment and we can work to turn off the body's response to the inflammation and repair the brain, there's no guarantee that once we get you better as a patient, you won't end up in another moldy environment and trigger the same cascade of events. That we can't fix. I think there are probably not good reasons, but reasons why we haven't seen the kind of public health response to this issue of sick building syndrome. Because unlike most other chronic illnesses, this is one where you can point a finger. It's the builder, the contractor, the employer, so the issue of worksite safety becomes real. And there are potentially culpable individuals or groups. And we see this, that there have been lawsuits where families or groups of families sue, the landlord or the employer or, the building contractor, and they win. But still, it's a field that's in its infancy in terms of being accepted as a clinical reality.
It's certainly more in its infancy in Australia. I looked up
even
one
more so.
person that was teaching about it.
Yeah. I know there's a recognition here, but for us, the great revelation has been, yes, mold is an issue, but there's a variety of exposures that turn on the same immune response and create the same symptom complex and damage the brain and so on and so forth, that it represents a whole disease entity. Mold is only one trigger of many.
Yeah. I wish I had about 6 more hours with you, but I'm going to try and ask you 2 last quick questions.
Sure.
The first one is, I'll ask them first. The first one is, do you think we're anywhere near measuring biological resilience, which you mentioned in your talk?
We are. There are actually a series of studies looking at the constellation of markers that together might indicate resiliency. So we're getting closer. It's as much a mathematical challenge as it is a one of measuring itself. And it's one of the reasons why we've moved to genomic work and transcriptomics, meaning the RNA expression of genes, because we're interested in why some people don't get as sick. And there is resiliency built into our systems. But usually researchers don't think like that. We're beginning to ask those questions. One of the other research studies I'm involved in is with U.S. Navy SEALs. And we know that some of them experience a traumatic brain injury and become very, very sick, permanently ill, and others can experience the same set of events and they don't. So I work with several different military physicians where we're collecting genomic data, proteomic data, as well as unique markers from the brain to try and identify why is it that some people, some Navy SEALs, are able to absorb multiple exposures, concussive events, lack of sleep, stress, toxins, and they're fine and their brain is fine. And then there's another group that never recover.
So because I'm only going to ask one question, I'm going to ask it in a way that I can put 2 things together. So you mentioned the importance of transforming growth factor beta and its control of interleukin-17.
Yes.
I'm really interested in the work of Dan Littman and Avalio Ivanov with regards to segmented filamentous bacteria and this interesting programmer, if you like, of the infant immune system. How does that play out? And where can people learn more about this? Because we've only given them a snippet, the tip of the iceberg. Where can they learn more about your work?
So I think as a framework for discussion, probably the website survivingmold.com is where a lot of this research is contained, where we house our studies, as well as all of the definitions of what these markers are, including TGF-beta1, which is, you raise it and you smile, but you have to understand there are 20,000 studies in the literature on TGF-beta1, and it's growing. So,
and it's growing.
And it's growing every day. Yeah, exactly. So we know that sometimes when that marker's high, it's regulatory and positive for health. And sometimes when it's high, it's not. And it's not when we see its impact on the pathological development of Treg cells. So Treg cells can become
hoodwinked.
Exactly.
Hijacked.
Exactly. So when we see that alteration, it's a complicated and interesting marker and topic. We measure it, we know it's important, but we're still learning more about it.
Can practitioners access your work via published work?
Sure.
Published books,
they can search.
For instance?
Yeah. Well, I don't have any books per se, but a lot of it, again, is on the Surviving Mold website. The book is coming. I added to the list. Running the master's and PhD program and clinic and other work obviously takes up a lot of my time. Interestingly, where I feel the great deficiency is in the field, I argue that there is no definitive textbook. One reliable source of information that pulls these threads together in a sort of evidence-based and meaningful way. There have been good attempts, but to me, that work has not been done. If I were to do one thing in that regard, and it's just my sensibility, I would, and we're talking about this at George Washington University, of beginning to put together that tome.
I, for one, certainly look forward to that publication. It is an absolute pleasure to meet you, finally.
Thank you very much. Yeah.
This is FX Medicine, and I'm Andrew Whitfield Cook.
Hi, this is Stacy, the baby maker Robert. I have put together more than a decade of my clinical experience into developing the first online mentoring program that deals with the ever-growing area of natural fertility. My Babymaker Network Mentoring Program is an online interactive program where you will learn how to address all aspects of fertility issues. If you are ready to be a part of an atmosphere that helps you build your practice while helping couples build their family, I look forward to getting to know you in the Babymaker Mentoring Program. Please go to bioceuticals.com.au and click on the Education tab for more information and to register.